So our next speaker is Bob Foy, and Bob is the director of the Kodiak NOAA Lab at the NOAA Alaska Fisheries Science Center. And he works on causes and effects of elevated CO2 on marine species. And I have to say, Bob is the kind of guy that usually has about 50 projects going on at once, and is also the person that shows up when the electricity goes out at 2 a.m. to his lab to make sure all these crab are still alive. And he's going to tell us more about these projects. So let's see.
Okay, thank you very much, Darcy. Appreciate you getting the OA network up and running, and for the overview and herding the cats and keeping us going. This is wonderful, much needed in the state of Alaska. And thanks, Jessica, for the presentation from afar as an intro to get us into what ocean acidification is. So, as was said, my name is Bob Foy. I work for the National Marine Fisheries Service at the Kodiak Laboratory. And I'm going to walk through some of the research that we've been doing, mostly in the laboratory, and then the ramifications of that for fisheries in the Aleutian Islands in particular. So there's a lot of principal investigators involved in the work I'm about to show you, lots of folks engaged since about 2006 on this kind of research, and you'll see on the side of most of my slides many of the scientific publications that are going into this presentation. So if you are interested in any of the details and a little bit more information, just let me know, because I'm certainly just skimming the surface of what we've been doing for the last decade. So I'm going to walk through how the Alaska Fisheries Science Center went about determining what species we were going to work on, and then moving into the funding end of our research. So our focal species are commercially important fish and shellfish species, and that's obvious because of the National Marine Fisheries Service mission. The prey, calcareous plankton, and shelter, in particular the corals. And what you're going to see is that most of what we've been able to focus on to date is the first dot and the third dot, mostly because of what we can do in the laboratory. And we have a long way to go at looking at the ecosystem aspects of ocean acidification and on the prey resources of the commercial species in particular. Our major objectives were to ocean pH monitoring, to support some of the work that Jessica mentioned before, to understand species-specific physiological responses. That's what I'm going to speak mostly to. And then to forecast population impacts and economic consequences. You're going to hear a little bit about that in the following speaker. We've also done some of this work by taking the data from our research experiments and expanding them up to the population level. To put it in perspective. So how do we get going with something like this? This just shows a general pattern. Oh, you guys can't see the arrow. This just shows a general organization here from bottom to top with small scale processes to larger scale processes. And so we wanted to start on the bottom. We wanted to know what was the individual effect of ocean acidification. We could guess what's going to happen at the population level if all the juvenile crab disappear. We wanted to know, well, what is the effect on an individual crab before we scale it up. So we started looking at individual tolerances. We look at physiological limitations. So we take animals, small fish and crab, and see how much pH they can take over a period of time. Growth and development. So that's something that we've been looking at in the laboratory because we're good at looking at it in the laboratory. We can measure growth. So that's something we can compare with this new perturbation and see if there's any impacts. Maturation, that's fecundity. How many eggs does a female crab have, for instance? Mortality. Then we start working up into larger scale, population level indices such as reproduction. And these are items that we can then take and model in our population dynamic models. And then lastly, dispersal. We've got a number of studies right now where we've got tagged crab in Norton Sound and Bristol Bay. We've got about 100 crab out right now with satellite tags on their back looking at movement patterns and dispersal that we can then relate back to environmental variability. So large-scale processes that we're looking for relative to climate change, range expansion, that's something we can do with the surveys that we heard Wayne talk about earlier. We can look at the changes between years. Life history changes such as growth, reproductive timing, habitat changes, and species interactions and larval drift. With specific to ocean acidification, we're looking at that mortality and recruitment, growth changes, calcification, so how much calcium does a crab have in its shell, for instance, and behavioral changes. So before we got down the path of taking these animals in the laboratory and playing mad scientist, we had to determine what is it about ocean acidification that we're really interested in. And often you hear that ocean acidification is only about the ability of calcium and carbonate to bind and the ability of an animal such as a shellfish to use that calcium carbonate. But we're also seeing in the literature now that ocean acidification, because of the lower pH, is affecting animals such as fish directly and affecting their behavior. It's affecting some fishes in Australia's ability to smell prey, for instance. So that's the pH aspect of ocean acidification. So there's two things going on, and I'm going to show two chemistry equations. That's it. I promised to cut the rest out of the talk. But basically all this shows is that carbon dioxide and carbonate bind together to create bicarbonate. So when carbon dioxide that you heard of in the previous presentation, 30% of that anthropogenic human-made carbon dioxide is going into the ocean, it's being bound to the carbonate that's in the ocean. So why is that important? Shellfish and corals need that carbonate to bind with calcium in order to make their shells. So if the carbon dioxide is binding to it, it's not going to be available for building the shells. It's as simple as that relative to the shell building. So again, you heard about this relative to the omega values. An omega value greater than 1 is supersaturated, meaning that calcium and carbonate will precipitate. If it's undersaturated, calcium carbonate wants to dissolve unless there's a biological reason or energy being put into the system to help keep it together. So with acidification, you're not going to see crabs rotting on the beaches. You're not going to see things just dissolving in front of you. It's going to happen at a slow enough pace. What you're going to see is effects on the population. And that's what we've been working on in the laboratory. Some hope. There's a lot of negativity in these presentations. So, and I'm going to talk about some of the realities of what we're finding in the laboratory, but there are a lot of animals, corals, coccolithophores in particular, that can use this bicarbonate, the top equation there. They can use the bicarbonate in order to bind and create their shells. So there are animals out there that will be able to acclimate and over long periods of time potentially adapt to the changes we're seeing in acidification. The question is, how will the commercial species that we're interested in adapt or acclimate? So in light of looking at the difference between pH and carbonate availability, for crab species, that's what is going to be the majority of my focus, we're interested in which may be affected by pH, which is affected by carbonate availability. So if you look at distribution here, this slide shows red king crab, blue king crab, and golden king crab, different life stages and the depths that they occupy. So to the left, you see that most of the red king crab and blue king crab are on the shelf in shallower waters, whereas the golden king crab, a brown crab is what it's called in the fishery. We know that that's a deep water fishery. They're found in deeper waters. So what you would expect is that it's possible that as ocean acidification increases in Alaska, what you're going to see is that pH is a more important factor for these shallower water species, and the availability of carbonate may be the issue for the deeper water species. So that's how we set about defining our experiments when we first began. More recently, we've started looking at tanner crab and snow crab and something similar, where it's possible that you would expect that tanner crab are potentially more resilient to changes in pH because they're in a shallower environment that sees the pH swings all the time, whereas a snow crab may be less tolerant because it's in slightly deeper water, but water that's more stable relative to changes in pH for now. So what we had to do next is biologists become chemists. So we had to take a step back and figure out how we were going to emulate what happens in the environment in a laboratory setting in a meaningful way. So we started out with some crazy experiments with hydrochloric acid, and we put carbon dioxide into car buoys and dragged them around the lab, and poor students trying to put projects together. But eventually we worked with the chemists, folks like Jessica, and we put together a system that's pretty complex, although extremely stable. So what we do here, and I'm not going to get into these details, but we take carbon dioxide, we bubble it into seawater, we have a super acidified system, and then we have a number of instruments and a number of probes that send all that acidified water into our different treatments where we hold crab. So basically what we do in the experimental tanks is we hold crab over a period of time at ambient pH conditions. We hold them at a pH of 7.8, which we expect to get in about 50 years on a global average in surface waters, and then a pH of 7.5, which we expect in 80 to 100 years. And our goal is just to see how do these things change over time. So we've got these flow-through systems. We're able to control the pH, and then there's lots of aspects of the chemistry cycle that we measure. So we started this project in about 2006 or 7. We finally dialed in the chemistry for some meaningful experiments in 2010. We worked through red king crab adult females. I thought that was my cue to dance. So that makes an ocean acidification talk more cheery. So we were working with adult red king crab females. We did a couple of studies that were just short 2-3 month period studies. We then started working with embryos and larvae because really where the majority of the influence of the environment on most of these species is during juvenile development, during those early life stages. So that's where we wanted to focus our efforts, and we've gone through that with red king crab as well. More recently, we finished a 2-year study on southern tanner crab juveniles. The reason this is important is most of the studies in the literature, it's difficult to hold an animal for a long period of time. So you see experiments for 2, 3, maybe 6 months over a short period of their time. But in order to understand the effects of these environmental perturbations, you really need to take them through the most stressful times in their life. When they're molting, when they're mating, when they're trying to hatch or metamorphose. So we were able to do that with tanner crab first over a 2 full year period where we held, I'll describe it here in a moment in a little more detail. We then looked at golden king crab adults. We haven't done this a lot, but in some respects they may be the canary in the coal mine and or the species that tells us where we could go in the future because they're already living at depth. They're already living in a corrosive environment. We weren't able to get into that with this presentation, but if you look at the waters in Alaska and what Jessica showed you up in the Bering Sea where there are some corrosive waters at depth, in the Gulf of Alaska at depth, they're all corrosive below 100-200 meters, somewhere in that range. Meaning that you go below that depth, it's already a corrosive situation because of natural earth processes, because of the upwelling of the world's oceans in the North Pacific. So you've got golden king crab living in this environment already. So how are they able to do it? How are they able to withstand a commercial fishery? And the question is, will that depth shallow and affect other species in the Aleutian Islands? That's what we need to look at. More recently, we've been looking at snow crab. We just finished a 2-year study on snow crab, so we're just getting into the results there. And that's a pretty exciting study. So what are we looking at? Simply, we first start looking at survival. Do these animals survive these treatments that we're putting them in? And we're always relating them back to a control. So I always get the question, well, of course they don't do well in your laboratory. You grabbed them from the bottom of the Bering Sea, you put them in a tote, you took the water away, put them upside down in a box, shipped them, sat on the tarmac at the Anchorage airport, and then you sent them to the Kodiak laboratory and put them in a blue tank. You wouldn't do well either. So we know that we have those issues associated with doing laboratory research, but we always compare it to a control. So we have animals that are in these control tanks that went through the same perturbations but are not in the acidified treatment. So that's what we're looking at. We also look at fecundity, morphometrics, how do crab grow, and then calcification, what kind of calcium content is in their shells. Lastly, we've been able to look at hemocytes. So hemocytes are the blood cells of crab and hemolymph. We've been able to look at their ability to protect their immunological functions. We've been looking at genetics. So is there any change in genetics, what we have found is that when you put a crab in an acidified situation, different proteins are expressed to try and protect the shell, and the shell under the shell called the cuticle. So when a crab molts, you've got that secondary shell, and it's putting all its energy into trying putting calcium and carbonate into that new shell. Population dynamics, and then bioeconomic focus. So this is a busy slide, so I'll walk you through it. But in essence, this is the Tanner crab study. So in 2011, what we first did was just bring in female wild-caught crabs. We put them in acidified condition, and then we allowed the larva to hatch, and then we looked to see what the condition was of the larva in these different treatments. In 2012, we brought those females in, we left them in the treatment, we then took their larvae and put those in treatment. So we had a crossed design where, for instance, a female that was held at 7.5 for 9 months, her larva was then put in an ambient situation in a 7.8 situation, and then a pH of 7.5. So we were able to run this experiment for 2 years. We then mated those females and ran the experiment again. So the difference there is now you've got the female putting all her energy into egg development before the larvae hatch in an acidified environment. It's more realistic. It's more consistent with what the animal is going to experience in the wild, as opposed to just looking at one life stage. And this will be important why I'm hitting on this so heavily here in a moment. For the experiments, we looked at a number of different response variables that I've mentioned. And then the key here were the carryover effects. What does that mean? Animals that were held for a long period of time. So if I'm a juvenile crab and my mom experienced no acidification effects, I don't have that history of stress and that history of condition being affected by an acidified environment. But if my history shows an acidified environment, so if the moms were exposed to acidification, the larvae were exposed, then as a juvenile crab, I've got that entire history to deal with those stressors when I hatch into the environment. So that's what we were looking for with these experiments. So what did we find? I'm not going to get into the extreme details of the project, but this is a good graphic to show the difference between year 1 and year 2. So the first set of pie charts on the left are year 2012, year 1, and then the second set is 2013 or year 2. The lighter color part of the pie chart means they hatched viable larvae. The larvae look good. Anything dark means that it wasn't viable or they were eggs that didn't hatch at all. So what you can see is that in the first year, there's really no difference between the ambient environment, a pH of 7.8 and 7.5. So when we finished the first year of the project, we said, ocean acidification is not likely an issue for Tanner crab. They're probably more resilient because of their environment. It wasn't until year 2 where we exposed the full life stages of the crab that we were able to see some differences. So you can see here that 87% of the larvae hatched just fine in ambient conditions. But when you go to the bottom at a pH of 7.5, over half did not hatch or hatched or were dead after they hatched. So 50% of the crab in that acidified condition did not survive. So again, that shows the importance of understanding the entire life history. So next I'm going to show some results for red king crab. So I'm just going to list some of these. Overall, we found that a decreased pH is associated with smaller eggs, smaller embryos, and larger yolks. So at first we thought larger yolks. This is the energy that the embryo uses as it grows, as in any egg. So we thought, well, this was a good thing. What it was, unfortunately, because it was smaller, the larger yolk was the animal's adaptation to try and put more energy into keeping the animal alive. It's a stressful period of time, so they had larger yolks. They had the larval calcification increased, and that suggests that they're putting all that energy from the yolk into developing the shell. It's going into calcification. The morphometrics were varied. The key here is that survival decreased. So all this energy that they're taking to survive, they're just not able to make it. There's just not enough energy in order to get through this life stage. We saw that juvenile growth was decreased. Calcium content really didn't change in the juveniles, meaning they're putting a ton of energy into their shells. But again, we saw decreased survival over time. So one aspect, I'm just showing a couple of the very specific projects here. One aspect of the red king crab work, we looked at shell mechanics. So this shows, this picture here on the left-hand side is the width of a shell. So you can imagine how small that is. There's an inner side, the endo side, the exo, and the epicuticle. These are all parts of a shell. So what we were able to look at using a number of techniques and instruments is the hardness of those different parts of the shell, the thickness, and then the mineralogy of those different parts of the shell. So this slide shows the hardness. We looked at the carapace, which is the back of the crab. We looked at the chela, which is the claw. You can see the outer part of the shell on the left-hand side graphics and the inner part of the shell on the right-hand side. And I know that's difficult to see, so I'll focus here. If you look at the left-hand graphic, you can see that the bars go down. And what that shows is that the outer part of the carapace on their back is becoming softer and softer as acidification increases. And you can see that also in thickness. It's becoming thinner over as you increase acidification. So what this one shows is, this is looking at the chela. So what this is showing is that there's not much difference in the thickness of the chela. What we did, however, find is that the hardness of the chela decreased with a lower pH. So what that suggests is that the chela is going to break down more often. The chela's job is to take bivalves and shells and break them. If the bivalve, or if the crab chela is more brittle, it's not going to be able to feed itself. If it can't feed itself, then it can't deal with this stressor and the energetics, as we just discussed, that it requires in order to get past the survival issues. So some folks have asked, well, what happens when you increase temperature as we expect and you decrease pH with ocean acidification? And what we find is that they work together negatively to affect the survival of red king crabs. So what this shows on the far left, the lightest dotted line, the faster it goes down, the more mortality that we're seeing. So what this shows is that in the warmest temperatures, the lowest pHs, we're seeing higher mortality. And the darker lines on the right-hand side of the graphic show that they survive longer if they are not acidified and if they're in cooler water. So this is one of our experiments. So again, that this suggests that over a 200-day experiment that these two factors, temperature and pH, are going to work together in a negative way with juvenile red king crab. So what are we doing with these data? The data are important in and of themselves, but what does it mean at the population level? So we've worked with a number of colleagues to take the laboratory data and develop functions that fit into existing population dynamic models. So what we've done here is we've taken the data on how many eggs are produced in the experiments in acidified condition. We've said how many juveniles will survive from the laboratory experiments. Now we take that and we put it into a population dynamics model. What does that mean for the stock in the future? So we take those data, we put them into a brand new pre-recruit model. So that's a model that we predict how animals are going to grow through time, how many crab are going to live from today to 7 years from now. We then take that model and feed the existing population dynamic post-recruit models. And those are the ones that currently we use to manage the Bering Sea fisheries. We then provide those to the bioeconomic models that are also currently being used to estimate the effects of fishery changes in the Bering Sea. And we've been able to predict changes into the future in both the economics as well as the population dynamics. I'm not going to get into the detail of these results because of they're fairly long and complex, but in a nutshell, for both tanner crab and red king crab, what we've predicted is that once the Bering Sea reaches roughly a pH of 7.8, there'll be a 20-year window where we really don't see the effect of acidification, just due to the natural variability. Everyone who fishes crab knows there's a natural variability or a pre-existing variability because of environmental conditions in recruitment. But after that 20th year, you see a precipitous decline in the recruitment of red king crab and tanner crab in the Bering Sea. based on our laboratory studies. So what we're looking at is 70 to 100 years from now, once the Bering Sea reaches this pH, then you start to see this mortality. Before, you likely see these issues associated with the Bering Sea crab fisheries. So does this mean it's definitely going to happen? Absolutely not. This is based on laboratory experiments. What this likely is a worst possible scenario. These animals may acclimate, they may adapt over time, and that's what we're going to talk about here in a moment. So very briefly, I'm going to talk about ground fish studies that have been done. So these have been done by Tom Hurst at the Newport Laboratory. He's looked at northern rock sole and walleye pollock and continues to do these studies. Northern rock sole he found to be more sensitive to low pH situations. You can see in the right-hand graph here that as carbon dioxide increases, the mortality rate increases as well. So he's seeing a higher mortality rate. Right now he's working on studies to look more into the mechanism of that. When you look at walleye pollock, you see something very different. There was no effect in changes of carbon dioxide on the survival or growth of walleye pollock in an acidified condition. So that's very good. What that means is that the initial impacts of acidification are unlikely to directly affect pollock. The questions now, and based on some other studies around the world, are there any behavioral effects? So that's what Tom's looking at right now. He's looking at cod and pollock to see whether there's any behavioral changes associated with acidification on those species in particular. So we'll see where that goes. So last couple slides here just to put this in perspective. This just shows the Aleutian Islands Area O fisheries. I've got the east and west of 174 line here, which delineates the golden king crab stocks. So you've got red king crab stocks around Petrel Bank, Adak. Here in on Alaska, you've got golden king crabs stretched along the entire Aleutian chain. So what do we expect? Golden king crab are already in an acidified situation. So the question is, will changes from human acidification change any aspect of the life history of golden king crab? Unfortunately, we know so little about the life history of golden king crab. We really don't know whether or not there's going to be an impact. But it's unlikely that we're going to see very much change with golden king crab because of acidification. With red king crab, however, there's lots of evidence to suggest that there'll be an issue. These stocks are already depressed, which means they're already in a state where they're not able to reproduce and replenish their stock. So this is yet another stressor that they're going to have to deal with as these numbers decrease in the North Pacific. So conclusions, changes are coming. Which ones are going to be more significant are difficult to tell. Crab are very sensitive to both changes in temperature and pH, as well as changes in the calcium carbonate saturation. And groundfish behavior may be affected. We still have a lot of work to do on that. So shellfish fisheries sustainability, can crab and fish resources acclimate? And there's some hope. There was a study done at the University of Alaska on Dungeness crab that did not find an effect of acidification on dungees. They're also very near-shore species. They're already living in this variable environment, so it's possible that they've already adapted over time to these kinds of changes in pH. There's some hope there that if it happens, if acidification happens slow enough, then the commercial species will also be able to acclimate through time. Lastly, some projects going on. I mentioned golden king crab, red king crab. We're finishing up snow crab. Red tree coral, probably the most important biological habitat species in the entire North Pacific because of its biomass and importance for a lot of the species in the Aleutian chain. A lot of the rockfish species, mackerel, use red tree coral for habitat, for protection. So we're raising red tree coral right now in the laboratory in Kodiak. So we've got them in different acidified situations. So we just started this in January. Hopefully we'll have some results in this next year. We're looking at both survival, we're looking at calcium content of the coral itself, and we're looking at the reproductive viability of the coral polyps through time. Walleye pollock, I mentioned this before, we're looking at scent detection and looking at multiple stressors at once. And then red king crab, A lot of the work we've done up until now has been a static environment. We bring them in and we put them at a pH of 7.5. The reality is the environment is changing diurnally, meaning every day. It's changing seasonally. So we're taking that and we're now putting that into our treatment experiments so that we've set kind of the threshold at what could happen. Now we're scaling it back to some more realistic situations. Lots of people to thank, and thank you for listening.